Hello, everyone, and welcome to this live broadcast from Logos Research Associates. I'm Rob Stadler. I'm an author and a scientist in the medical device industry, and I'll be your host for this presentation. Logos Research Associates is a group of scholars and scientists that are committed to upholding a high view of science and a high view of Scripture. We sincerely believe that scholarship, logic, and the scientific method all show that the Bible's historical claims are credible and ultimately are superior to the secular model of origins. That is, the origin of the universe, the origin of first life, and the origin of all kinds of life. Logos sponsors many creation research initiatives and projects involving many different experts and scientific disciplines, all working toward the goal of glorifying God by exploring his creation. The goal of this live stream series is to provide you with the opportunity to hear from top researchers who will present cutting-edge scientific evidence for the creation account. And before I introduce our speaker for today, I'd ask that you please join me in a short prayer. Heavenly Father, we thank you for this time together, the opportunity to study your creation. Lord, through your creation, we find the process of science coming from your creation. And it gives us opportunity to learn from your genius, from your creativity, and it ultimately points to you. We thank you for that. We pray that you'd bless our speaker this evening. Please give him clarity and the right ways to present to the audience. We also pray that you'd bless the audience, Lord, that they would be open to hearing and learning and growing closer to you through this process. And we pray in Jesus' name. Amen. So our speaker this evening is a good friend of mine, Dr. Joe Deweese. Welcome, Dr. Deweese. He is a professor of biochemistry, a director of undergraduate research at Freed-Hardeman University in Tennessee, and he's also a world expert in a particular family of enzymes, which are essential for all of life. Every aspect of life has to have them, and they are known as topoisomerases. And I'm calling him a world expert, and that's true because he has 28 peer-reviewed manuscripts on this one topic. So for all of you who have had these burning questions about topoisomerases for months and weren't able to find your answers, tonight is your night. Dr. Deweese has a BS degree in biochemistry from Freed-Hardeman University, and then he went on to get a PhD at the prestigious Vanderbilt University in biochemistry. And then he went on to be a professor of pharmacy at Lipscomb University for 12 years, and then recently has transferred back to Freed-Hardeman University. So welcome, Dr. Deweese. We look forward to hearing and learning from you.
Thanks, Rob. I appreciate it. Appreciate you introducing me and starting me off. You set the bar high. So, but I hope everybody can learn something about topoisomerases tonight and see why these are such cool enzymes. So, yes, thank you, Rob. Appreciate it. So, as we get started tonight, we want to talk about topoisomerases. These are amazing enzymes. You're going to learn how to say the word, hopefully, before we're done tonight. But also why these things are important and why we care about them. As Rob mentioned, I do my work at Freed-Hardeman University in Henderson, Tennessee. We're a small Christian university in West Tennessee. Hope you'll check us out and send folks our way. We're actually in the midst of our summer research program right now, and we've been doing that. This is our third year for that, focusing in kind of the biomedical and biochemical sciences. So excited about that program and the students that I get to work with there, and get to show them a little bit about why this enzyme is so cool. So we deal with topology all the time in different ways. Maybe it's Christmas lights or weed eater line or the pool robot cord here or an extension cord. Topology is something that we deal with in life in different ways. We interface with it. It's annoying at times when your maybe charging cable gets tangled up in the mix of other cords. It used to be that we would have headphones, right, that had cords, and those would get all tangled, but now everything is Bluetooth and wireless, and so that's kind of gone away. But the reality is we understand on a fundamental level that when you have a long string or strand of something, cord, it's easy for that to get wound up, intertwined, entangled. And I'd like for you to consider that that's not altogether that different from what happens in our genome and in our DNA. You know, if we think about all of the DNA in a cell, cells are measured, in at least human cells are measured on the order of micrometers, maybe 10 to 100 micrometers, but within that, the nucleus would be maybe 5 to 20 micrometers in diameter. So we're talking about millionths of a meter here, right? 5 millionths to 20 millionths of a meter. And there's about 6 meters worth, or excuse me, there's about 2 meters worth of DNA in all of that. So you kind of, you know, think about that 6 feet worth of DNA crammed into, something that's the space of 5 to 20 microns. So it's a huge level of compaction here to get that genetic information down into a really tight space. And that involves a number of key players of proteins to make that happen, including the enzymes we'll talk about tonight, but also includes things like histones and cohesins and condensins and a number of other proteins that coordinate different parts of what we call the scaffold. So we often think of DNA as this, right? The double helix, where you've got 2 strands that wind around each other. If you look at them on the end, they look like a circle. If you look at the side, you see this kind of waving, winding staircase kind of pattern. And that is true that DNA is a double helix, but in our cells, it's in a much more compacted form. And so we think of it as winding around, wrapping around. So here's what we think of chromatin as actually including a lot of proteins like the histones. So I have here colored in kind of brighter colors here, the red and the blue. Green and yellow would represent the histone proteins in the middle of DNA that's wrapping around them, kind of like a cord wrap if you're trying to organize your extension cord, for instance. Turns out that's happening on a massive scale in our cells. This figure from the NIH kind of depicts, as we think about, maybe you picture a chromosome as being this highly compacted form, but if you were to stretch all that out, what you'd realize is that there's this kind of solenoidal twist here that represents groupings of these histones all wrapped around each other, basically. And so here we have nucleosomes formed where DNA wraps around the histones to give us what we call a nucleosome. And it can exist in different states, either closed off or opened up, depending upon what's needed. And so that's kind of, a really fundamental question in DNA is, all right, well, how do cells access the information they need? Turns out we have a whole family of enzymes that we call chromatin remodeling proteins. And so to illustrate this, let me take you across campus from where I am right now to the what we call the ARC, the Academic Resource Center. And on the second floor of the ARC, there are bookcases. It's a library. So imagine that. We have books. And what you notice here on the side is that some of these bookcases are kind of close together. Well, it turns out they're on movable tracks, and you can push a button and you can slide the bookcase over and get in and access the books that are in the space that's currently closed off. And that allows for more space up there, right? So where those students are seated would not exist if we had to spread all of the bookcases out evenly all the time. And so instead, we save space by condensing some of those together and having kind of these movable bookcases. And I want you to think of the DNA as kind of doing something similar, where some of the DNA can be closed off or opened up, just depending upon what's needed at the time. And so that's a constant process going on in our genome. And so again, it underscores this importance that this DNA has to be really highly maintained to keep it from getting all tangled up. And so what proteins and what functions are at the root of keeping all of that from messing up? In fact, that's somewhere on the order of 10,000-fold compaction when you go from this double helix to the nucleosomes to the solenoidal twist all the way up into the actual chromosome. It's a huge level of compaction. So all right, so that's one issue, but okay, what about replication and transcription? So replication is when we copy the DNA and make another copy. Transcription is when we use that information in DNA to make the molecule we called RNA, or ribonucleic acid. So doesn't DNA need to be separated for replication and transcription? And the answer is, of course it does. And in fact, Watson and Crick even recognized this in one of their papers in 1953. They observed that since the two chains in our model are intertwined, it's essential for them to untwist if they are to separate. See, they recognized in their structure, if we're going to get these things apart, there's got to be some kind of untwisting. They say, although it's difficult at the moment to see how this process occurs without everything getting tangled, we do not feel this objection will be insuperable. Love the term there, right? We need to bring that word back. Apparently, it's underutilized. So what are they saying here? Essentially, they're actually using a form of what my friend Paul Nelson would call design triangulation. They're recognizing that there's a problem that if their model is correct, this problem has to have a solution or life wouldn't exist. And they recognize that. And so they were essentially predicting the existence of things like the topoisomerases. Let me illustrate the problem here. If you just take a rope that is twisted up in a double-stranded fashion right here, and you start pulling those ends apart, what happens beyond that spot? Well, you're going to get kind of some compression of these coils. And remember, chromosomes are quite long, so you could say, well, wouldn't the end just untwist? Well, no. Imagine a rope that is a mile long and maybe a centimeter in diameter. And if you start at any one point and start pulling those strands apart, you're going to get some compression and supercoiling to result from that, because it can never reach the end. It's never going to get out there all the way to the end and uncoil, even if it's unobstructed. And so we get what we see as supercoiling. You can do this at home anytime. Make a double-stranded rope segment, attach one end to something that's fixed in space, a doorknob or a handle or something, pull the ends apart until you get a lot of compression of the double helical nature there, and then stop pulling it apart and allow the rope to kind of pull the ends kind of closer together, and you'll get this effect here, this supercoiling, which is trying to relieve this torsional strain of the compression. And that's a reality. This is happening in our genome. So as we take DNA and we try to copy it with replication machinery, we're going to get supercoils ahead of that process building up. And so these things have to be dealt with. This is a real thing. So how do we deal with the compaction? How do we deal with the separation? What's keeping all of this from getting so knotted and tangled? So here are the problems. Replication and transcription give us supercoiling. Processes like recombination and repair can cause knotting of the DNA. Replication results in linked chromosomes that have to be resolved prior to mitosis. And so we call those catenanes, and that has to be undone. If each of these represents, let's say, a separate chromosome, like 2 copies of chromosome 2, for instance, we've got to resolve those so that those get separated out and we can move on with mitosis. So how do we deal with these things? What is the answer? So it's not insuperable after all, as they predicted. We have enzymes that were named topoisomerases. So topo for topology, and isomer in chemistry is a molecule that has the same components but a different structural relationship. And then that suffix there, ase, just means an enzyme. So it's an enzyme that can alter the isometric nature of a particular segment or molecule, in this case DNA. So we call them DNA topoisomerases because they basically can remove or alter topology or topological strain in these molecules. So for instance, the supercoiling we mentioned a minute ago, that can be relaxed by way of topoisomerases. And I'll explain the type 1 and type 2 here in just a moment. Recombination and repair can unknot or can be unknotted by the action of topoisomerases. And then these links or catenanes can be unlinked by topoisomerases. You notice here there's two different types, type 1 and type 2. These are kind of broad categories. There's actually subfamilies within these groupings. But the big picture is that type 1 versus type 2, they both can alleviate supercoiling, but the type 2 is going to be more involved with your unknotting and your decatenation. And part of that is because type 2 involves a mechanism that can actually break both strands of the double helix. And that's not why it was named type 2 initially. It's actually because of how it changes the topology. But it turns out that the type 1 enzymes cut one strand and the type 2 enzymes actually cut both strands. So here, for instance, here's type 1A and type 1B. Type 1A, when the enzyme, I'm just using a little Pac-Man symbol here to represent the enzyme, it will attach to the 5' end of the DNA and allow for alleviation of torsional strain. Type 1Bs attached to what we call the 3' end, and the 5 and 3' numbers have to do with the actual sugar molecule in DNA, the ribose or deoxyribose in DNA, and the position on that sugar that we're talking about. The type 2 enzymes, you notice, can cut both strands. There's a 2A and a 2B. And so we have the ability to cut with a 4-base pair stagger or with a 2-base pair stagger between the cut sites. So the type 2 enzymes and type 2A and type 2B enzymes are multimeric enzymes in general. While some of the type 1s have more than one protein that's part of the functional enzyme, the type 2A and 2B have 2, 4, or even 6 proteins that come together to make up those structures. So here's a type 1 topo over here versus a type 2 topo where the two halves here, one is colored and one is in gray. So you can kind of see the contrast between the two halves. So we have these different families. Turns out there's actually even another one. There will be a test on this, so go ahead and make some notes here, right? Type 1A, 1B, and 1C, 3 different families of topo 1. And then there's type 2A and 2B, 2 different families of type 2 topoisomerases. So this is where it gets kind of into the weeds of the details, because you might say, well, this requires a topoisomerase. And we would say, well, what kind, what type, what family? And this is where you get into the various details. Some of these are present in eukaryotes. For instance, humans we have 2 type 2A enzymes. We call them alpha and beta, topoisomerase 2 alpha and beta. We also happen to have 2 type 1A. We call them topoisomerase 3 alpha and beta. And we also have a topoisomerase 1B and a topoisomerase 1B mitochondrial. So we have 6 different versions of topoisomerase in us, and there's actually a couple other proteins that are seemingly very similar as well that kind of fall into a related category but have more to do with recombination. So this just gives you an example of some of those different proteins. Notice many of them require magnesium, some of them require ATP. They have different mechanisms. So the topo 1 enzymes if it's a 1A, it will cut one strand past the other strand through the break and then put the strand back together, just a single strand break. The type 1B and 1C will make one break and then kind of rotate part of the DNA, swivel it around to alleviate the torsional strain. The type 2 enzymes, 2A and 2B, create double strand breaks and then will pass an entire double helix through the break in the other double helix. Another interesting note that I want to point out here is that there are also viral topoisomerases. So there's 1A, 1B, and 2A viral topos, meaning there are viruses that have their own topoisomerases that have to be present to help maintain their genomes, which is kind of interesting to consider because that kind of is an interesting question when you think about the origin of topoisomerases, if they're even required for certain viral genomes. That's very interesting. It really implies their integral nature in nature as a whole. So as far as we know, all life forms need topoisomerases. And what's interesting, as I pointed out a few minutes ago, if you caught this, the type 1 enzymes, pretty much all enzymes, all organisms have a type 1 topo, but the type 1 topos, turns out, are not the ones that actually are absolutely critical for life. It turns out that type 2 topoisomerases Because of that function back here, in unlinking the sister chromatids after and during replication, this function right here, if it doesn't happen, cells don't divide and organisms die. And so for that reason, we think of type 2 topoisomerases as being really essential to life. There's some thought that maybe some type 1 enzymes could facilitate that on some level, and that may be true in some cases. But by and large, we know if you knock out the type 2 topoisomerases, the organism or the cell will not survive. And so that's pretty interesting to think about. And because of their integral nature in the function of the genome of each of these organisms, it turns out that we can actually disrupt this function and hijack that for therapeutic purposes. So one of the reasons anyway, other than that they're really cool, one of the reasons that topoisomerases are studied is because targets of both antibacterial and anti-cancer drugs. In other words, there are drugs designed to target the bacterial topoisomerases that we can use to treat humans and rid us of bacterial infections. So that's drugs like the fluoroquinolones. You also have anti-cancer drugs that can target human topoisomerases and kill cancer cells. It's been estimated that about half of all chemotherapy regimens include a drug that targets some form of topoisomerase. So they're very widely used in the anti-cancer field. There are also inhibitors that are being looked at for blocking viral topoisomerases. So that's another way that these may be used for the treatment of disease. So very interesting family of enzymes, or multiple families here of enzymes, and a lot of diversity. Another quick note before I move on from this table, this type 1C topo 5, the last I read, this is the only known instance of that enzyme. So it's thought of as kind of an orphan gene or orphan protein in that sense. Orphan in the sense that it is kind of one of a kind. It doesn't have like a precursor to it. So from an evolutionary perspective, there's some explaining to do as far as where that popped into existence from. So you can see all these different structures here. Of course, they're in really pretty colors, but these are just some of the examples of the diversity of different topos. The one in the bottom middle we'll come back to a little bit later, but that's actually a bacterial topo 2, where you can see the DNA kind of wrapping around on the ends, that kind of extension part there in yellow on one side and kind of blue or cyan on the other. What I want you to get the picture of is that topoisomerases are sensing topology. They're sensing the three-dimensional shape and structure of the DNA. And we'll talk about that a little bit more in a minute. Now, how do they actually carry out these functions? What's kind of at the heart of that? And one of the things that I think is really cool is that pretty much all of these topos use a very similar mechanism to cut the DNA. You see, when they cut the DNA, they don't just cut it and let it go. They actually cut it and grab onto one end. So I pointed that out a little bit earlier in one of the slides where I said it attaches to the 5' or the 3' end. But it turns out that essentially all topoisomerases have an active site tyrosine residue. This is a diagram made by some collaborators of ours years ago in one of our papers, where they were showing that this is kind of the theorized process by which we think the tyrosine attacks a phosphate. You get a transfer of electrons, and you ultimately get this phosphate attached to the tyrosine of the enzyme. And then you have a free end of the other end of DNA right here. And this is happening twice, right? So this is happening on one strand, and then a few bases away happening on the other strand, the type 2 topoisomerases, so that you get a double-stranded break. One other thing I'll point out, we'll come back to this in a little bit. You notice these different colors. So black here is the DNA. The green is amino acids from one half of the protein, and kind of the bluish color is amino acids from the other half of the protein. So one of the cool things about type 2 topos is this active site, this region where this is occurring. It's a very three-dimensionally constrained arrangement, requires amino acids from both halves of the enzyme to come together. So you get these mirror image active sites composed of amino acids from opposite protomers, as we call them, of the enzyme, which is really, really cool. So type 2 DNA topoisomerases, again, we have 2 in humans of the type 2A form, alpha and beta. What's the difference? Well, alpha seems to be more involved in replication and mitosis. Beta is more of what we consider a housekeeping gene. It's present all the time, appears to be really involved in transcription and in chromatin remodeling. Remember the moving of the bookcases. So topo2 beta is more of kind of a maintenance protein. Alpha is more specifically involved in replication. And for that reason, actually, in my particular interest in it in the research lab is to look at topoisomerase 2 alpha, because that would be the isoform we really would want to target from a cancer perspective. Turns out most of the drugs that we have, essentially all the drugs that we have that are clinically approved, really don't differentiate between the two. They hit both of them. And so you get some of the off-target toxicities and side effects resulting from that. So again, these make a double-strand break. There's a 4 base pair 5' overhang attaching to the 5' end of the DNA. Requires magnesium and ATP. Untangles the DNA strands during replication, transcription, mitosis, repair, all of these things. And again, cancer drugs are used to target these, but there can be off-target effects from that. The domain structure, so if I just took the protein and laid it out flat here, so the color coding kind of matches over here with the exception of this CTD, it's not found, and I didn't put the pink or the light blue. But you can see up here, the ATPase domain's up at the top in yellow, followed by the transducer in orange, top rim in red, and then this DNA binding domain leading down to the C gate down at the bottom. Again, this CTD structure is not present. The other half I just left in gray. It's kind of on the back side of this particular image. So you can kind of see where the different parts of the enzyme are and where the different components are. If we kind of look at a couple different views of it here. So you kind of look at the side here is the DNA in green in the middle. This is the DNA that's actually going to get cut and separated. When you turn it, rotate it, you can see that there's another kind of cavity through here. That's where another piece of DNA is going to be and get passed through that break down into this lower cavity. And then this view is kind of looking down from the top on top of that. We think of topoisomerases as having kind of a gated model, a multi-gated model. There are different ideas around this, a 2-gate versus a 3-gate model. Generally, I think the type 2A topoisomerases, you can argue, have this kind of 3-gate model going on where we think that there is at the top, the ATPase domain back here would represent what we would call the N gate, where N stands for amino terminus. So when you think about a protein structure, let me get back to the slide here. The amino terminus would be one end, the carboxy terminus or C terminus would be the other. So when I say N, I'm talking about kind of the front end. C is talking about kind of the back end. And so the N gate would be up here at the top with the ATPase domain. The middle with the top rim and the DNA active site would be called the DNA gate. And then the C gate would be at the bottom. And the DNA that gets transported through, we call the transport segment or T segment. The DNA gate segment, we call simply that, the gate segment or the G segment. So what that looks like then would be this. Here is our topoisomerase. Here is DNA coming into the active site. The G segment gets heavily bent right here. And the gold segment here is going to be bound in the top part here by the ATPase domain. Somewhere in this process of bending, the enzyme is able to actually make the double-stranded break, but doesn't necessarily pull those apart immediately. We think that the ATP, based upon kinetic evidence, that the 2 ATP molecules are not hydrolyzed at the same time. It's kind of sequential. And so with the hydrolysis of 1 ATP, we think that the gate can actually open and the transport segment can be pushed through the G segment. The gate segment gets closed, and then the hydrolysis of the second ATP allows release of the transport segment, and the enzyme can then reset and release the gate segment. So it steps through this process of kind of bending and cleavage, binding to both segments, separation of the gate segment, transport segment pass through, putting the gate segment back together, and then releasing both of them. Again, there's kinetic evidence that maybe hydrolysis occurs sequentially. One of the mysteries in the topoisomerase field is, what is the ATP actually doing? So in this case, in the type 2A topoisomerases, it's thought that the energy of ATP hydrolysis is not needed to drive, for instance, the passage of the transport segment through. The thought is that that actually may be driven just by the strain, the torsional strain of the DNA itself wanting to be relieved. And so it's not thought maybe that the ATP is powering that, but maybe the ATP is helping keep this in-gate together so that you could get strand passage and the whole thing not fall apart. Because remember here, in this particular case, type 2A topos are two separate proteins, and each one is going to connect to one end. So what's keeping those two proteins from just falling apart in the middle of this process? Some thought is that maybe that ATP hydrolysis is keeping those ATPase domains together to keep all that from falling apart. So here's just a quick animation to illustrate what this can look like. So this is the topoisomerase animation. This is actually out on YouTube. You can check it out. Just search for topoisomerase 2 animation and you can watch the whole thing. This is just a clip from it. But what you can see is that we're going to have a double strand break here in the middle of the screen, and we've made one double helix clear just for a moment. We're going to pass the strand through that break, and then we're going to put the DNA back together, and then we can the thought is maybe there's some potential rotational, there's some structural evidence for maybe a rotation event there, and then release of the transport segment from the bottom, and then resetting of the whole process. So it's pretty cool to think about what this thing is doing. It's able to make this double-stranded break. It's then able to take the other segment, pass it through that break, and then put the DNA back together. It's a pretty fascinating thing. So this thing, like, imagine if your shoelace was knotted and you could undo the knot and put your shoelace back together. Like that's what this thing is doing, right? This is the capability of it. So it's really, really amazing to think about what these machines do. So as we think about that process, this is what we call the catalytic cycle. It's a cycle in that this thing can go round and round and repeat itself over and over again. And that's what it's essentially having to do in your cells, is to repeat this process just time and time again. And so it's really fascinating. And we can actually monitor this with DNA and gels. I mentioned this in my talk last month, that we can actually look at a molecule of DNA, a supercoiled molecule of DNA that's all twisted up, and we can actually monitor its progression from twisted up to what we would call kind of the relaxed state of DNA up here at the top. And in between, we have all these different intermediates that we call topoisomers. So this process is fairly simple, but requires topo 2, requires ATP, requires magnesium. You got to have the proper pH. There has to be enough salt present, and temperature. I didn't put on here, but the human enzyme likes to work at body temperature. And so that's also required in this process. So the question on your mind perhaps is, okay, well, how does the topo 2 know where to bind? How does it know where to go? Is it like a restriction enzyme? So those of you who are familiar with restriction enzymes, they cut very specific sites, oftentimes palindromic sites. So you can read the sequence here, AA, GC, TT, and then if you read it down here, AA, GC, TT. So it's a palindromic sequence, and turns out Hindi3, this particular enzyme, can cut at those particular sites. Well, that's not what these are like. It sees topology. It sees the twisting. I made the kind of a mirror image of this with the thought that topoisomerases can discern whether something is underwound or overwound. I didn't have a good way to depict both of those, so I just mirror imaged it here. But the idea is it could be twisted tighter, positively supercoiled, or it could actually be twisted back the other way and slightly underwound. And that will also result in supercoils as well. And we call that negative supercoiling. So you can actually have both directions. And if we go back to our structure of the bacterial topo earlier, I just point out this idea that notice how the DNA wraps around on the outside here. There's some evidence that wrapping and the directionality of that wrapping combined with any other strands that are involved in this, that's what the enzyme is really seeing or looking for, so to speak, in its topology. And I apologize, this is poor quality image here, but taken from a paper I wrote years ago with my mentor. But you actually can quantify the wrappings. If you think of a Mobius strip, like a strip of a piece of paper, and you twist it a couple times and put it back together, you can actually make a molecule that's either negatively supercoiled or positively supercoiled using this kind of node convention here. And then you think of topoisomerases as taking a molecule that's like this and actually removing one of these by crossing the DNA over itself. And then you end up with a molecule that is now no longer supercoiled in that way, but rather can essentially be relaxed. And so that's kind of the concept here. So how big of a deal is this, though, when we think about DNA? Well, let me illustrate using a plasmid we use all the time in lab. There's an enzyme, PBR322, very common plasmid. We use it in our experiments very regularly, where we can monitor its progression from supercoiled up to a relaxed state. And we can actually measure how many, if you counted these bands, you could actually measure. There's about 11 topoisomers represented here. And each time topoisomerase 2 does its work, it changes the linking number by 2. And so we call that a delta LK of 11 times 2, or 22. If there's 4,360 base pairs in PBR322 and 10.5 base pairs per turn, that gives us what we call a linking number of 415. If you divide those two numbers, the delta LK by the LKO, you get a sigma value of 0.05, which essentially means that this plasmid is 5% underwound. I actually should have put a number or a sign on this, because the delta LK in this case is negatively supercoiled, so that should be a negative 22. But it's 5% underwound. Turns out genomes are approximately 5% underwound globally. As far as we know, most genomes are in this state of about 5% underwinding. What does that mean? Well, scale it up, okay? This is a 4,000 base pair plasmid. Scale that up to, let's say, the size of E. coli, right? 5 million base pairs. So take that number of interlinks here between it and multiply that out essentially by a factor of 1,000, right? So you're talking about 22,000 interlinkages along the chromosome of E. coli. So E. coli require topoisomerases to deal with these 22,000 interlinkages at that 5% underwound state. And so this is a real problem. This is a real challenge for organisms to deal with, all of the twisting that takes place. Now, the thought is this underwound state that we find genomes in, the benefit of that is thought to be that it kind of slightly opens the double helix, makes the double helix a little bit easier to open and to access by, let's say, the helicase or the RNA polymerase for replication or transcription, respectively. So there's some thought that that's why it gets to that slightly underwound state. Well, then, of course, once you open it up for replication or transcription, you're going to start getting positive supercoiling or overwinding, and that has to be relieved as well. Lots of different control points to consider here. Okay, so the functional enzymes in the case of eukaryotic type 2s, type 2As have two halves, so you got to have both halves. There's a question of localization. There's a question of post-translational modifications, protein-protein interactions. Lots of important questions, and many that we really don't have great answers to. But let me ask you to consider the functional enzyme requires both halves, and this is for several reasons. One is this is a reversible covalent bond between the enzyme and DNA. So remember, the enzyme's cutting and ligating. So this little structure down here in the corner, the green Y782 is the active site. It's attached via a phosphate to the backbone of DNA. So there's a broken piece of DNA right here. And that's a reversible. bonds. Remember, I call this the reversible molecular scissors here. Ligation appears to be the preferred state. What do I mean by that? I mean that the enzyme doesn't tend to like to stay like this. It doesn't just grab on, cut, and hold. It tends to actually prefer putting it back together. For instance, when we run experiments in the lab where we're just trying to measure how much DNA is cut, we have to add a vast excess of enzyme to be able to see anything, and without any type of cancer drugs or adding anything else to the reaction, we only get about 1% of the DNA cut at any one time. So it's a very low, tends to prefer to put the DNA back together, which is kind of honestly what you want in an enzyme like this. You don't want something that's chewing the DNA up. You want something that prefers to put it back together. Note that the cleaved DNA can only be ligated and released once that G segment closes. In other words, that gate in the middle, if it's open, you can't let go of the DNA. So that's a really neat kind of control point there, that you have to have closure of the gate segment in order for you to get ligation and release. And then you've got this 4 base pair overhang, which is nice because it kind of zips the DNA back together before you get ligation, which may help prevent aberrant ligation with, sections of DNA that don't match up. And so this kind of helps ensure proper alignment and gets those back together. We also know that we've got to move this thing around. Turns out there are, interestingly, both nuclear export sequences and nuclear localization sequences. So there's both a way to get the enzyme into the nucleus and out of the nucleus. It's not totally clear why there are 2 NLSs and 2 NESs, but there appear to be 2 of each. So some redundancy there. Maybe there are other binding partners that could block some of these sites from time to time. But there are apparently 2 different sequences. And I think there's some evidence that, at least for the NLSs, one of those is stronger than the other. But nevertheless, there still appear to be 2. So we have nuclear localization sequences, which would bind to the import proteins, for instance, like the importins, which would grab the protein and take it through the nuclear pore into the nucleus so it can do its job. Remember, in human cells, this thing's getting made out in what we call the cytoplasm outside the nucleus. So it's got to get shuttled into it. Without those NLSs, it stays in the cytoplasm and it can't do its job. So it's got to have those to get in there. We also know there are a ton of post-translational modifications. In fact, this screenshot is from Phosphocyte, which maps different phosphorylation and other modifications onto the protein sequence. So all the little lollipops that you see along there represent sites where a modification has been identified. And the height of the lollipop is how many times that's been referenced in papers in the literature. So you can see some of these have been very well studied, especially out here at the C-terminus. And we're going to talk more about that here in just a minute. But you can see they're present all across. But what is interesting is that about half of all of the modifications, if we totaled all these up, about half of all of them occur out here at the C-terminus. And so file that away. Remember that, because that's going to come back in a minute. But you can see the color coding too. Many of these are phosphorylation. There are a few acetylation, quite a few ubiquitylation. So that's, remember, ubiquitin helps target something for destruction, sending it to the proteasome. So lots of modifications. We did a study several years back looking out here at the C-terminus, and we actually kind of tried to study just the modifications that had evidence from basically from being modified during mitosis or in association with mitosis. So you see all these sites on topoisomerase 2 alpha that get modified by mitotic kinases or modified in association with mitosis. Many of these have assigned kinases, but some of them do not. So we're not always sure exactly what modifies all of these, but it does appear that there are a lot of modifications that occur out here in association with mitosis, which implies that these must have some functional consequence on the enzyme, the fact that there are so many. Another interesting thing is, what does topoisomerase 2 interact with? Does it interact with other proteins? There's a database out there if you're bored and just really curious. The biogrid.org is a database of protein-protein interactions. And you can search for your favorite protein. Everybody's got one, right? Search for your favorite protein, and it will give you a list of proteins and even small molecules that your favorite protein interacts with. Turns out topoisomerase 2 has well over 120, 130 plus potential binding partners. Many of those overlap between topoisomerase 2 alpha and beta. So you have this kind of overlap in the middle. But you've got dozens upon dozens of different proteins here that have been implicated. Now, most of these only have very minimal information about the interaction. In other words, it may just be that they were found in a particular experiment or screen, but there's really not biochemical data to say exactly where they interact. So the field is wide open at the moment as far as trying to track down some of these interactions. There are a handful that we know where they interact, but most of them we don't. And so that's a really interesting question out there. And how many of these are real? How many of these are relevant? But even if, let's say, this list is off by, I don't know, let's say it's off by 10 or 20%, that's still a ton of possible binding partners present here. So, unless you're just going to discount the list as a whole, there's something really interesting going on here. A lot of different potential binding partners. Maybe some of these are second order. Maybe some of these don't interact directly with topo. Maybe they interact with something else, and that is interacting with topo. That's possible. But needless to say, there's a lot here to be explored. Many of these proteins make sense. They may relate directly to topo function in some way, and so some of them you look at, you say, yep, that makes sense. We understand why that would be on the list. Others are pretty puzzling and have no clue why they're on the list, and it'll be interesting to see how that plays out. All right, there's also some other interesting questions. How does, for instance, when DNA is binding and ATP is binding, how is it communicated between, let's say, this upper part of the enzyme, the ATPase domain, down into the middle? How is that all communicated? Well, you may have heard me refer to the orange part as the transducer domain. There's some thought that amino acids in this orange region help the core of the enzyme know when the ATPase domain is in a particular state or that it has ATP present. So there's some thought that there's communication going on. The nature of that is not yet clarified. There's still a lot of questions around that. But even recently, I saw a paper on molecular dynamics simulations showing that there is evidence that when you affect something up at the top there in the yellow part, it can affect things down into the enzyme. In fact, we have some bioinformatic evidence that that's going to be the case as well. So there's also coordination. You've got strand passage with ATP binding, hydrolysis of ATP, the sequential nature of that. So there's kind of an asymmetry. You've got this symmetrical enzyme, but you have an asymmetrical, catalytic process taking place, which is also very interesting. And then you have opening and closing of the gate region and all of the things that go along with that. So that's pretty cool as well. Now, what about the C-terminal domain? I've mentioned it a couple times. Here is kind of this is the original alpha fold structure of topo2. And you see the big squiggly line going around the gray thing. The gray part would be the part we've already been looking at, the structured region. The orange part would be this C-terminal domain that's highly flexible and disordered. We call them intrinsically disordered. I know that I've got some friends that really don't like that particular phrasing, but the idea is that on its own, it's not folding into a nice, neat, curly Q kind of shape. And so why is that? What is it doing? This is the region in orange where half of those post-translational modifications of the whole protein take place. This is where we've got several known binding partners. And so I think that evidence probably indicates, and I think others would agree with me, that this part probably takes a much more defined shape in the presence of the right binding partners. In other words, think of this as like a flexible, think of it as like a hand in a glove where the our hands are not in one conformation. They can actually shape themselves around whatever we're binding to. And I think that's probably what's happening here. It's not meant to be a rigid domain. It's meant to be something that's flexible and can bind to things. So that's pretty cool to think about as well. For those of you who have kept up with AlphaFold, it now can actually fold what we call dimers and multimers, where you have more than one protein present. And so I actually fed this into the AlphaFold 3 server, the sequence of Topo 2. I told it to use 2 of them, and out popped this nice, neat structure. And for the most part, it looks fairly good through the core middle of the protein there, nice and structured. But you notice we still got the squiggly on either side outside. And so that's very interesting. The squiggly line seems to indicate that there is, still a lot of uncertainty in the algorithm as to what to do with that. And that makes sense. I think it's probably because we haven't found the right binding partners. And with AlphaFold 3 server, you can actually feed in other proteins and see if you can get this to take a shape. And there is some preliminary evidence that we can actually get it to take a shape. So, but I'm not going to disclose all those secrets just yet. So you'll have to come back another time to hear the rest of that story. So what roles does it play in the activity of Topo 2? Can we exploit those roles? So the work of my lab right now, as I kind of wrap up here, is we're looking at that C-terminal domain region and asking the question, all right, could we actually target that region therapeutically, maybe to shut it down in the case of cancer? Because one of the interesting things is that region, the C-terminus between alpha and beta, is very, very different. And so that's an interesting thing. If they're different enough, could we selectively target one and avoid the other, and maybe avoid some of the off-target side effects and toxicities of some of the cancer drugs? So we published our first paper on mutants back in 2021, ACS Omega. We have a software tool that I developed along with Kirk Durston and Tim Wallace, and the software work was done by an outstanding software developer, Thomas Townsley. So when I say I did this, they did this, right? Some really super smart people. And our software tool is able to predict interactions, or at least interdependencies, I should say, between different sites in a protein. And we're using that to look at the C-terminal domain and other regions. And we published some of that work within the last month in another journal, as I note down here. So we are moving forward on this. We're trying to do real science. We're trying to really look at this carefully and think about what are the functions, what's going on out here in the C-terminus. Can we catalog those functions? Some of these mutants from my original study we're actually still using. We've got experiments planned for tomorrow on some of them. So we're still working on some of these, trying to understand what they're doing. If we messed them up, why are they messed up? And trying to apply this to the bigger picture of understanding what's going on out there in the C-terminus. So really fascinating enzyme. I really think it's cool. These are the reversible molecular scissors of the cell. And they're really, really fascinating what they can do and how they kind of maintain the topological state of the DNA. And so, as we kind of get ready to wrap up here, what do we make of all of this? These are amazing little machines at work in your cells, keeping your DNA from getting too tangled. We've learned that we can hijack them for the purpose of the treatment of disease. And I'm always fascinated by the mechanism. The more I study, the more I look at it. And the more I think it points to, the Romans 1 verse 20 concept, the more I think it points to the need for design and a designer, because it really underscores just how much planning and engineering had to go into developing life. This is a real engineering problem in our cells that has a real solution that we get to study and explore every day. So thank you for hanging out with us for a little bit tonight. I hope I didn't put you to sleep, but I'm really excited just to spend a little time, and happy to open it up to questions and comments and thoughts as we kind of wrap up here. So thanks everybody for being out there. I appreciate that. And we'll get ready to kind of open up into the discussion session. So thank you all for being out there. Okay. Let's see, Rob, I've got a,
Can I get, oh, I'm on, okay, I'm back.
Okay.
Yeah, wow, totally amazing, Joe. I'm so impressed. And what a privilege it is to just sit back and have a world expert on a topic like this, just give us the basic education here. And to think, folks, just to give you some appreciation for this, to think that Joe has put 20 years of work into studying basically this one enzyme. And that's not to say that Joe is slow in understanding things. It took him 20 years. We're
Right.
talking about, something so complicated like this, that you have a group of people, not just Joe, but a group of world experts that are dissecting this thing and understanding how it interacts and how it works. And 20 years of doing that, and we still have many, many questions, as you saw, of what's really going on here in just this one enzyme. So just imagine the complexity of all of life with many enzymes like this one. It's just fascinating. So like
Yeah.
Joe said, we invite you to answer, to put in your question. Go ahead, Joe.
No, you're right, and it's, there are experts all around the world. In fact, during the pandemic, uh, uh, one of our colleagues out in the UK set up a Zoom call for topoisomerase researchers around the world, and that was a really cool thing, just to kind of connect everybody in a time where everybody felt so disconnected. And it reminded me of how big the community is of folks that study this one kind of area of enzymology and biochemistry, is just looking at topoisomerases, and people specialize in these different ones and study them and try to understand them and explore them, and it's really a fascinating thing. And I'm humbled and honored. I did not know that I would keep researching once I got out of grad school. Frankly, I was kind of done with lab work. When I finished grad school, I was burnt out, and my department chair at Lipscomb gave me an opportunity to do a research project about a year into my time there at Lipscomb. Said, "Hey, take a student, do a summer project, see what happens." And that was kind of the start of my lab back in about 2010, 2011, and we're still rolling, and we've been incredibly blessed. So yeah.
And you've got some job security, because there's still a lot to figure out here.
Yeah, there's a lot left. That's
So
for
let
sure.
me start off with a question, Joe.
Okay, sure.
I think, as you know, I have this great interest in the origin of life.
Yes.
And those who imagine that life began through purely natural processes, they like to use the term proto-life,
Mhm.
that early on there was much simpler life than we have today. And so, we see that these topoisomerases are essential for all life that we know of today. So no life could live without these things. Even, as you said, some viruses have to have them. So would it be possible for some sort of proto-life, earlier form of life, to live without the topoisomerases, to not have that complexity? And if so, how small would a genome have to be in order to survive without these things?
Mm. Yeah, that's a great question. So let's make a couple assumptions here for a moment, and let's assume that original genome was, let's say, double-stranded in nature. The problem gets really big really fast. So I illustrated with just a 4,000 base pair plasmid that you're going to have 22 topological linkages when you're at kind of the standard 5% underwound state. And any opening of the double helix is obviously going to perturb that and mess with that. So, fairly even small molecules, need this to be resolved. Otherwise, it's a major issue. So you take yourself to, kind of the smallest genomes that we're aware of, they're still on the order of 400, 500,000 base pairs in that range. So you're talking about a lot of potential topological linkages. So, I mean, is there a scenario where maybe they were all separate molecules and much smaller? Maybe. But, some of these proteins are fairly large, and the number of different proteins you need multiplies out by the number of different, genes to make those proteins. And now you're starting to talk in the tens to hundreds of thousands of base pairs at a very, very minimum. Really several 100,000, honestly, to get many of the proteins that we know are needed for life. So there's a lot of theorizing around where that began, especially as it relates to the fact that there are some viral topoisomerases. That discussion is out there that, okay, well, maybe these originated in viruses. In fact, what's interesting is there's some argument that these originated in viruses and then came into eukaryotes. And then there's other evidence suggesting maybe they were in eukaryotes and viruses borrowed them. So there's even debate around those questions. And I was reading a paper on that today, and they basically came to the conclusion we can't totally tell which direction it came from. We think it was this direction, but there's also evidence for the opposite. So yeah, that's a great question. I think you would need an incredibly small genome, and it would be so small you wouldn't have the proteins that you need to carry out life.
So you're saying something like 4,000 base pairs. If it was one DNA strip of 4,000 base pairs, you'd probably need a topo to keep that going.
You need something because if you're going to make a copy of that, it's going to get intertwined, and you're going to have positive supercoils. You're going to have catenanes. Those are going to have to get relieved in order for that thing to get copied. And those people out there who've done like PCR-based mutagenesis, where you use the polymerase chain reaction process to mutate a plasmid, they're aware that even in those settings, you have to have certain enzymes in place to get that to work, especially if your plasmid is 8,000, 10,000 base pairs or 15,000 base pairs or more. And so there are ways that you can on a small level make extra copies of something, but at some point you've got to put that in a cell and let the cell carry it out and separate it out and so forth. So yeah, it's a good question.
Yeah, so the people studying the origin of life have a lot of explaining to do then, if they can imagine such a thing. And this relates to a question that came in from the audience here, that do evolutionists have guesses as to how these things could have developed?
Yeah, that's a great question. So one way that some researchers look at it is they look at it in terms of the different parts of the protein and say, okay, well, this looks kind of like something we see over here, and this looks like something we see over there. So if I step back a slide here to this, make sure, okay. So the top part, the yellow part, ATPase domain, that domain, that particular fold, it's hard to kind of picture it here because I'm not spinning it around in 3 dimensions where you can see it from all angles. But that particular shape and fold actually is shared with other enzymes that have ATPase domains, such that it's even got a name. It's a GHKL ATPase. And so the argument would be, well, maybe this ATPase was borrowed from some other enzyme that had already developed that. And then in the middle there in red, the top rim domain is found in topoisomerases and enzymes called primases, which are involved in replication. And so maybe that top rim domain had a particular origin and was borrowed. And so that's kind of the direction that this goes is, well, this part looks like this and that part looks like that. So maybe it's just we've borrowed these modules and stitched them together to get this, functional structure. That's a hard, in my mind, that's a hard case to make. That, well, yes, these modules do appear to have very good similarity to similar modules in other proteins. To argue that somehow those were just stitched together in a long evolutionary process, that kind of begs the question of what's even possible there. And let me step back one further, because it's not just a question of the origin of, let's say, topoisomerase 2 or topoisomerase 2 alpha in particular, but you've got to explain the origin of a bunch of different topos. Okay, so this is 5 different families, and within these families, we could break it down further, and I could make the case that there would need to be somewhere on the order of 7 to 10 independent origins of topoisomerases when you really kind of break into some of these groupings. And so topoisomerases would have to have evolved 7 to 10 different times independently, many times landing on some of the same structures and features, but distinct enough that they had to be kind of their own origin.
And that's because there's not much homology between these different ones?
Yeah, there's separate mechanisms. There's different within their sequences. There's very low sequence similarity among many of them, and so they might have a very similar fold, but an entirely different or almost entirely different sequence, just depending upon the type or the version. So there's a lot of interesting questions there about who has what. I was reading today, for instance, the archaea organisms, which tend to be living in extreme conditions and things. They have this really neat enzyme we call reverse gyrase, which can actually actively supercoil DNA, which is pretty cool. So it does some neat functions. It can actually make it positively supercoiled on purpose. Well, that's found in archaea. Turns out archaea don't have normal type 2A topos like topo 2. They do have, some of them have a gyrase, which again is kind of a subtype of type 2A, but pretty much all of the archaeal organisms have a topo 6, which is a type 2B. So they have this kind of whole separate, version over there. And there are a few eukaryotes that also have that. But there's not a lot of similarity between the type 2A and type 2B in terms of, just straight sequence identity. There's a lot of conceptual homology as far as how they work, but they're entirely different proteins, which is pretty cool.
When we consider the kind of just so story, right, that evolution would need to produce in order to come up with where topos came from, one thing I can imagine is that it's relatively easy to come up with an enzyme that breaks apart DNA, right? If you had a random enzyme generator just cranking out random things, it wouldn't be too hard to get a destructive enzyme.
Yeah.
But that, of course, would be destructive. And so maybe the first thing you'd come up with would be something that just rips everything apart. But getting something that can tear something apart for a useful purpose and put it back together, now you're talking difficulty there.
That's a great point. And if you think about it, I mentioned in the talk, there's biochemical evidence that these enzymes prefer to put the DNA back together such that the equilibrium is shifted toward ligating rather than cutting, where you might look at other enzymes like restriction enzymes, and they're very rarely are they going to put it ever back together. Once they cut it, it's cut and it's done. But these enzymes tend to prefer to put it back together and, in fact, stay covalently attached until they can put it back together. And here's another layer, Rob. Not only are these enzymes designed to do that and to put it back together, but if something goes wrong and the topo gets stuck, there are another family of enzymes that we didn't have time to talk about tonight that actually come in and can get the enzyme off the DNA and release the DNA from it. How fascinating is that? So we have, in fact, we have an engineered response that can remove the topo from the DNA in a way that tries to preserve the DNA and allow it to get put back together properly. That's pretty cool. So, and that's a pretty neat story in and of itself.
And going a little further with that, I'm guessing that the restriction enzymes, they just go around cutting up DNA, I imagine them to be a lot simpler than the topo. So it's easy to come up with one of those and easy to cause destruction, but hard to have an enzyme, like you said, that prefers to put stuff back together again.
Yeah, that's true. Now, I haven't studied the structures of a lot of restriction enzymes, but that in general, they're a bit more simple than this because they're not having to hold on to the DNA and manipulate it while passing DNA through. It's just a matter of getting in and cutting it. And so they tend to be much smaller enzymes and on a comparative scale, much more simple for sure.
And precision is of the utmost importance then in putting things back together the right way.
That's right. That's right. Absolutely.
One question from the audience, you used the word discern about these molecules, that they can discern what they're doing. But how does a molecule discern anything. It's just a machine.
Right, right. So understand that, we're using what anthropomorphic language here to describe, biochemical realities. So this structure I was showing earlier, if I could, and I don't have it pulled up where I could do this right now, but if I could show you this in 3 dimensions and rotate it around, you would see that there's a directionality to the DNA and the twist of the DNA such that it fits just right in a particular angle. And I think that's probably generally true broadly of topoisomerases, that there is a sense in which if you get DNA at the right bend, the right twist, then it tends to fit well with the enzyme. And so in that sense, we think of it as discerning between, kind of the handedness of the coils of DNA. So I mentioned earlier this idea of negative supercoiling and positive supercoiling. So picture in your mind a rope that's negatively supercoiled would be underwound, like you're kind of almost separating the strands. Overwound rope would be positive supercoiling. And it turns out that the supercoils made as a result of that are twisted in opposite directions. And what we know is that certain topos have a preference for positive versus negative supercoiling. So in that sense, we think, and I think it's a 3-dimensional structural thing where if it's twisted one way, it's going to fit around the enzyme well. If it's twisted the other way, it doesn't fit around the enzyme. And so again, I think this picture I thought was a good kind of illustration or concept for us to think about. It has to do with the 3-dimensional shape of the DNA as it twists in 3-dimensional space as to whether or not the enzyme is going to prefer to bind to it and act on that side or not. So that's a great question.
It sounds like something that had to be designed in order to do that.
It's pretty amazing, for sure.
So here's another question from the audience here is, these topoisomerases, they grab on and open up the DNA and relax it. But how do they know where to grab on and where do they clip the DNA? How do you know?
Yeah, that's a great question. So as we think about it again, remember it's not a restriction enzyme, so it's not a sequence oriented per se. It is about this kind of shaping or twisting of the DNA in 3-dimensional space. So there's a couple things going on here, though, that we need to clarify. One is that topoisomerases are really very abundant in the nucleus, and so there's a bunch of them. And I think of them as kind of being stationed all throughout the genome. Okay, so these things are just all over the place. And they can associate, for instance, topo2 alpha, we know can bind to one of the histones, actually more than one of the histones. And so I think of it as kind of being stationed along the DNA in different places and kind of ready to act at a moment's notice. And so, it's able to kind of find or sense, so to speak, the 3-dimensional shape of the DNA. And if that shape fits, then it's going to bind to it with a certain degree of affinity and then start the process of actually cutting it. So it's not a sequence specific thing. I don't have a good illustration of this, but I could show you in their papers where this has been published. If you take that plasmid we talked about earlier, PBR 322, if you take that and cut it up into segments, like 500 base pair segments, you can actually map along that segment where a topo will cut. And what's interesting is it will reliably cut at some of the same spots. But then when you analyze those sequences, it's not like it prefers necessarily a particular set of nucleotides, but it does tend to go back to the same places over and over. And we see that in our genome too, which implies that there's some level of organization of where the topos end up on our chromosomes so that they're in the places where they're needed probably the most, which is kind of cool to think about. So they seem to get stationed at the same spots or in the same regions repeatedly. So I think that probably has to do with some of the proteins from that list of protein-protein interactions I mentioned. I think there are probably proteins there that are helping recruit the topo and get it to a particular site and kind of hold it there. There's another concept, and this is kind of newer in the last, I'd say 8 to 10 years, this discussion's come up. Everybody, I think, is familiar with the idea that oil and water don't mix, right? If you put oil and water in a cup or in a measuring cup, you're going to get a layer where they just separate, and you got oil and water, just separate layers in this vessel. Well, it turns out that proteins can clump together in groupings where essentially they make not so much a layer, but almost a sphere in the liquid, where they're kind of in a different phase. They call that a phase condensate. And topos can apparently do that with DNA. And so they kind of enter this separate phase from the rest of the solution around them. And so topo in this phase condensated state, we think of as probably in a real high affinity state for the DNA, bound to the DNA really well. And that's kind of the state we think it gets stabilized in. Now, what mediates that phase separation is actually the C-terminus that I keep mentioning. So that disordered domain, that's really what's facilitating that. So when you think about site selection or where they go to, it's about recognition of topology is what it comes back to. It's about recognizing where on the DNA those particular events are happening, where is it needed, where is torsional strain the worst, and that's where topo is gonna act. So good question.
Yeah, I can see you have plenty of work left to do.
Yeah, there's a lot more.
I think I have one last question just
Okay,
from my own head here is, you've mentioned that these topoisomerases are targets for chemotherapy or for antibiotics. Is it as simple as like sticking a piece of chewing gum in a machine that you just kind of want to gunk up the work so that these things don't work, or is it a highly specified attack on a particular part of the molecule?
That's actually a great analogy. So the term we use for the most common mechanism that these drugs work against the enzyme is the molecular doorstop. So it sounds kind of funny, right? But the idea of a doorstop is what? You put it in front of the door and it keeps the door open. Well, we know that when topoisomerase cuts the DNA, there's a broken spot in the DNA. There's an opening. And drugs, for instance, like the drug etoposide can actually slip into that break and keep the DNA from going back together. And the term we use in our field, now this isn't popular among oncologists because nobody likes to think of this word being used, but we call these types of drugs topoisomerase 2 poisons because they poison the mechanism. They block it from being able to put the DNA back together. So in the technical literature, you'll read about topoisomerase poisons, and the idea is the poison is something that actually blocks it from finishing its normal function and kind of makes it stuck, just like you said, kind of gums it up in that sense. And so what happens is essentially when you have a toposide present with topo2, you're going to get a buildup of single and double strand breaks in your DNA because the topo is just going to get stuck on there and can't let go. And if you get enough of that, it overwhelms the cell and its repair mechanisms, and the cell's going to ultimately have to commit apoptosis, cell suicide, and die. So that's the idea. Now, if it's less than successful, you could potentially cause more damage. So we know etoposide, and I think the numbers are about 2 to 3% of patients with etoposide can develop a secondary leukemia a couple of years later. And so that, you might get cured of your lung cancer, but then later develop leukemia. So this, it's like, okay, what's worse here? And so while these drugs are very effective in many, many cases, they can also lead to some problems later on too. And so we'd like to figure out ways to avoid that, make these more useful. So yeah, that would be the main mechanism. There are some other drugs that work on the ATPase domain. There's only, I think, 1 or 2 of those. Maybe one of those is clinically approved, and it's used in very specific cases alongside a poison of topoisomerase 2 because it actually helps reduce some of the toxicity. So yeah,
fascinating, absolutely fascinating. Joe, I really appreciate you teaching us along this way, and I love learning from you. So Thanks again for an excellent presentation, fascinating topic.
Yeah.
Thank you, Rob. Yeah. So for everyone else, as a reminder, we want to encourage anyone watching to check out the Logos Research Associate website. And on that website, you can learn about the various research efforts that our associates are going into here, like Joe, find information about their publications, and also an opportunity to consider supporting one of these exciting creation research projects. And while you're on the website, you can also subscribe to the Logos email list so you can receive notifications about presentations like tonight, and also receive the Creation Evolution Headlines newsletter. And a recording of this video will be made available on our website. It'll be on YouTube, so you can share it with other people who've always wondered about topoisomerases, but just haven't met Joe yet, and they want to learn about it. Our next presentation is going to be on Wednesday, July 10th, and this is with another expert, Spike Saras, and he'll be presenting about creation and astronomy. So for those who are fans of the James Webb Telescope and all the cool images that are pouring in from the telescope there, I'm sure Spike will be there to answer your questions and give us updates on what we're learning. And I would like to now close us in a short prayer. Dear Heavenly Father, we thank you for this opportunity to learn about your creation and just to take some time to marvel at the complexity of what you've accomplished, the genius of what you've done, and to give you the glory for that. And we thank you for Joe. We pray that you'd bless his work, his research, and his efforts even to cure cancer and to make better antibiotics by working on this one single enzyme of your creation. And we thank you, pray in Jesus' name. Amen. So again, thank you all for joining us, and we look forward to seeing you in the next broadcast on July 10.
Thanks, Rob.
Thank you. Bye, everybody.